Hey, welcome to 6.2.1 specification if you want to have impulses. This is the first just pause in the nerve impulse. I've popped this cell here, a neuron, there just as a reminder of the different parts of the cell. So if you can remember that, then ignore it. If you want to stop and have a little look at that before we carry on, then by all means do. Okay, so resting potential. So every neuron, if it's not transmitting a nerve impulse, it's said to be at resting potential because all neurons are still polarized at rest. So we can refer to nerve impulses as electrical impulses as well. During resting potential, there's a positive charge outside of the axon and a negative charge inside the axon. So we're thinking about this axon, the long axon here of the nerve cell. During resting potential, the sodium-potassium pump is working. You can see that down here. For every 3 sodium ions that are being pumped out, 2 potassium ions are being pumped into the axon. And that's obviously using ATP because it's a pump, it's an active process. You often get 2 marks for just saying 3 sodiums out and 2 sodiums in for every pump. The membrane potential during resting potential is minus 70 millivolts. Now, when we come to look at the action potential spike later on, that'll make a little bit more sense, but it crops up here and there in exam questions as well. During resting potential, the sodium channels are closed. So this picture only shows you the sodium-potassium pump. There are also sodium channels and potassium channels in the cell membrane of the axon. Sodium channels are closed, so there's no other movement of sodium during resting potential. However, the potassium channels are slightly open, so the potassium ions are able to diffuse back out of the axon. Now, this means that because you're pumping 3 sodiums out for every 2 potassiums in, and some of the potassiums are then able to move back out via the channels, that's why you end up with a more positive charge on the outside, because obviously sodium and potassium are both positively charged, but you've got more sodium ions outside, and you also got some leakage of potassium ions. Okay, so the first part of the nerve impulse is depolarisation. Now, the stimulus has to reach a threshold for an impulse to be sent. So depolarisation is the first kind of first stage of the impulse. The threshold is about minus 55 millivolts. Again, when you look at the action potential spike later, you'll see that a bit more clearly. When we say that the stimulus has to reach a threshold, it means that whatever is causing the action potential to be fired needs to reach the threshold. So the stimulus needs to be strong enough to reach a threshold. For example, if you had your eyes closed and you held 2 hands out and someone put a feather in one hand and a brick in the other hand, you might not feel the feather because the stimulus isn't strong enough to fire action potentials and nerve impulses, whereas you would definitely feel the brick because the stimulus is strong enough for you to feel it. An impulse is just a wave of depolarization along the axon, so it's like a Mexican wave of depolarization. There should be an I in there. Apologies. During depolarization, the sodium channels open and sodium ions flood in. You get a mark in the exam for saying sodium channels open. You usually get another mark in the exam for saying sodium ions flood into the axon. During depolarization, the inside of the axon becomes less negative. So the charges flip. So previously, during resting potential, it was positive on the outside and negative on the inside. That then flips and it becomes positive on the inside of the axon and negative on the outside. There is quite a nice diagram later on to help you visualize this as well. The membrane potential increases to around plus 30 millivolts. Again, we'll have a look at that on the action potential spike later. And just a little tip there, if you try and bullet point your answers and avoid too much waffle, it makes it easier for examiners to find marks, and I think it just makes you a little bit more likely to get the marks. These two sodium channels open in here and sodium moving into the axon. Okay, that shouldn't be first. Repolarisation. So once we've depolarized the membrane, we then need to repolarize it. So during repolarization, potassium channels open get to your mark, and potassium ions flood out of the axon. This then helps to restore the membrane potential to resting potential. Again, you'll see it on the much anticipated action potential spike or graph that you're going to see very soon. And then we move to hyperpolarization. Now, during this time, we can also call it the refractory period, hence why that's here. Potassium channels close quite slowly, and essentially too many potassium ions flood out of the axon, and this causes the inside of the membrane to become more negative than it would be at rest. During this time, that section of the axon can't be depolarized, so it means that the action potential can't go backwards, it has to only go in one direction, forwards. Which is useful because otherwise action potentials could get stuck in the middle of neurons and just kind of ping backwards and forwards, and messages would never get to where they need to be. Oh, you can see here during the refractory period as well, the sodium potassium pump has restarted. Okay, this diagram here I just thought was quite nice. So you can see here you've got your area of depolarization. So obviously, at rest it's positive on the outside and negative on the inside. Here you can see that it's flipped. It's actually positive on the outside and therefore would be negative on the inside. As that wave of depolarization moves along the axon, so that section of depolarization has moved along. So the whole axon doesn't depolarize at the same time. It's just one area at a time, like a wave of depolarization all the way along. So once this next area has been depolarized, you've got this section here that is repolarizing. That means that your action potential cannot move back that way. So it doesn't want to move back that way. It wants to keep continuing to the right in order to get to the axon terminals here and pass on to the next neuron. We don't want your action potential pinging back to the cell body. That would be no use at all. And then you can see again here, as that wave of depolarization moves on, you've got this area is now repolarizing because it was depolarized here, and this has been restored to resting potential, ready for another action potential to arrive if needs be. These arrows here in the depolarization just show the fact that local currents are set up here. So the sodium ions will diffuse into the area that's in resting potential and help to depolarize them. Yeah, that's all that means. Okay, much anticipated action potential spike. So we have here at minus 70 millivolts, resting potential. So we can see that this axon is at a resting potential of minus 70 volts. Here, what's called the rising phase is the depolarization. Now you can see that there's also these that are labeled as failed initiations. Now what's happened is there's been some stimulus, but the stimulus hasn't reached this threshold of minus 50 millivolts. So therefore, no action potential was fired. You wouldn't feel these because no nerve impulses or action potentials are fired. At the peak of I had plus 30 millivolts on my slides, plus 40 here, but it's kind of about 30 or 40 millivolts. After that, it's then going to repolarize. So this is your repolarization. And then here, we talked about it becoming more negative during the refractory period or hyperpolarization, and then back to resting potential. So we've got resting potential, depolarization reaching the threshold, depolarization all the way up here. We've got repolarization, repolarization, hyperpolarization or the refractory period, and then back to resting potential here. Okay, this one first. So your wave of depolarization, during the refractory period, your sodium channels can't be opened, which obviously means that it can't be depolarized. This means that there's a delay between impulses, which is useful because it means that the action potentials never kind of catch up with each other and they'll never overlap, because we don't want that to happen. It's not possible for that to happen. We also need to understand the all or nothing principle. So we talked earlier about the threshold. So you either reach the threshold and the action potential is fired, or you don't. You can't fire kind of a small action potential or a large action potential. They're all the same size. Or the change in voltage is the same every time. The only difference is that if the stimulus is bigger, then the action potentials will be sent more frequently. So therefore, you would feel something more vigorously. Wrong word. You would feel it. For example, someone could just touch your face, and there would be infrequent action potentials. Whereas if someone hit you around the face, then there would be very frequent action potentials because the stimulus is bigger. Oh, missed that one. Sorry. Frequency of action potentials is limited. And then factors affecting the speed of a nerve impulse. I quite like to ask this question. So your neurons can be myelinated. Now, you should already know that you've got Schwann cells that surround your axon. Now, those Schwann cells kind of all come together to form the myelin sheath. If a neuron is myelinated, it means that the wave of depolarization can jump from here, and it jumps all the way over the Schwann cell to this node of Ranvier, and then here to this node and here. So it doesn't have to depolarize the whole axon. Therefore, it increases the speed of the nerve impulse. And this is also referred to as saltatory conduction. The larger the diameter of the axon, the quicker the transmission, because it means that there's less resistance to the flow of ions. They can move more quickly. The sodium ions can diffuse more quickly to the next section in order to help depolarize it. And also, increasing the temperature increases the speed, but only up until about 40 degrees C, which is obviously close to body temperature. And then after this, the proteins denature, just like you learned when you did about enzymes.